Welcome back, folks. Last time when we talked about exocytosis and endocytosis, introduced the idea of coating on vesicles, and we talked a little bit about Clathrin. So I'd like to get into that a little bit more here. A couple of videos in this topic. So let's get going. So what you can see from this table here is there's a variety of types of coated vesicles. So most vesicles that are involved in protein and lipid transport are coated in some way, and so they're called coated vesicles. And that's because of the layers of proteins that cover the cytosolic leaflet of the transport vesicle. And so these vesicles, these coated vesicles, they're involved in vesicular transport and vesicular traffic throughout the endomembrane system, as well as in the processes of exocytosis and endocytosis. Like you see from the table here, there are several types of coat proteins. We won't get into all of these. The most well studied are the Clathrins and then COP1 and COP2. So we'll talk about those over the course of these couple of videos. The functions of the coats are primarily twofold. One, they play some role in determining the destination of the vesicle, and they also contribute to the curvature that's needed for the vesicle to form, and they stop the vesicles non-specifically fusing with another piece of membrane. If you just had naked vesicles all the time in the cells, they could fuse with any old random piece of membrane they encountered, and so that stops the fusion occurring until the vesicle is close to the membrane that it needs to be close to. So let's take a look at Clathrin-coated vesicles. These are these vesicles are surrounded by coats made from two types of multimeric proteins. That just means that multi means obviously many, several, and meric means part or piece. So there are many pieces to these protein coats. The two main components here are the Clathrin molecule and then a variety of different adapter proteins or AP proteins. So the shape that the Clathrin proteins form and the way they assemble around the vesicle as it forms provide the driving force to turn a flat planar membrane into a vesicle, into a spherical vesicle. So the basic unit of Clathrin structure is called a triskelion. This is one of these wonderful words that we come up with every now and then in biology, a triskelion. Each triskelion is a multimeric protein composed of three heavy chains, like you see here on the left in red, and then three light chains shown here in blue, and you can see that the heavy and the light chains radiate from what's called the central vertex. These triskelions then assemble into the hexagons and the pentagons that you see in a lattice that forms around Clathrin-coated pits and on the vesicle surfaces. So you can see individual triskelions. Here's one in red, here's one in yellow, here's one in green, lots in blue, and so you can see how these assemble to form these complex multimeric shells, and it's that shell which gives you the targeting, it gives you the prevention of fusion with other membranes, and it induces membrane curvature. So let's take a look again at this assembly process of a Clathrin-coated vesicle that we touched on a little bit when we started talking about endocytosis. So here we're looking at receptor-mediated endocytosis. So here is a receptor in gray. The little green ball shows you the ligand for the receptor, and so the ligand will associate with the receptor. And then we have binding of AP complexes to the receptors and to some of the triskelion components. Now that requires some ATP and GTP, and it's the association of the Clathrin coat with the AP protein at the membrane which starts to cause this curvature that we're seeing in the membrane. Now initially these Clathrin coats are made from hexagonal subunits of triskelions, and then as more and more triskelions add, you start to see a mixture of hexagons forming and pentagons, and you don't need to worry about too much about why that would be. I don't really know, to be honest. And so we then start to see more curvature and invagination of the membrane. Then we have some association of dynamin, which we introduced a little bit ago in one of the earlier videos, and that's responsible for kind of constricting the vesicle so that we get this pinching off kind of mechanism. And so what happens is that the dynamin binds GTP, and when it hydrolyzes GTP, it kind of ratchets closed every time it hydrolyzes a GTP, and that ends up basically pinching the membrane off to produce the mature vesicle. So that budding there is called scission. That's the opposite of fusion. So and that separates the vesicle from the plasma membrane surface. So that's how you get vesicle formation through Clathrin coat assembly. So that process is kind of shown here in these first 3 steps, but you also need to recycle the Clathrin. You have to send the Clathrin back to the cell membrane so that it can be reused, so we don't get into these cycles of just using things once. And so there's a step after the vesicle is formed called uncoating, and that's an ATP dependent step. There's an uncoating ATPase which liberates the Clathrin from the vesicle, and that leaves us with a naked transport vesicle, and that can go off and fuse with whichever membrane it's being targeted to, and then the uncoated Clathrin can be recycled, sent back to the membrane, associated with AP proteins that are associated with receptors, and we can repeat the whole process. So that's a little bit about Clathrin-coated vesicles, and next time we'll look at some different types of vesicles. I'll see you soon.